Welcome back to the second session, focusing on other renal cystic disorders. And I am going to turn it over to the moderators to lead the session.
Thanks, Brad. I'm going to start off here. You took my punchline away to the second session. So we're going to talk about advances in other renal cystic diseases now. And I'm excited to introduce the first speaker, Dr. Michael Köttgen. Dr. Köttgen is a professor of nephrology at the University Medical Center Freiburg, Germany. There, he serves as the head of the Deutsche Forschungsgemeinschaft-funded PKD Center Grant and is a vice speaker for 2 nephrogenic center grants. His research focuses on the molecular pathomechanisms of autosomal dominant polycystic kidney disease and on transport processes in the kidney. And today he will talk to us about the role of the co-chaperone DNAJB11 in ADPKD.
Thank you, Katie, for the introduction, and thanks, Terry and the organizers, for the kind invitation to present some of our data on the role of DNAJB11 in ADPKD. So the work I'm going to present today was mostly done by Tilman Busch and Björn Neubauer, and consequently I asked them to present their data, but they denied and said I should do it, which tells you 2 things. One, I'm working with very humble individuals, but secondly, they are the ones who are really calling the shots, so you're looking at the real bosses of the Köttgen lab right there. So we're in the session of the other renal diseases now, and I have to admit that we had no strong interest really to study the other cystic diseases because we had our hands full trying to understand classical ADPKD. Today I'm going to talk about how we got into the other disease and maybe some interesting links between these other diseases and classical ADPKD. So for us, this all started with trying to understand the function of the polycystin complex, which you've heard a lot about already. They form PC1 and 2, form a receptor ion channel complex. And we still have a rather limited understanding about how this complex is activated and what the downstream effectors are, although we heard something about that yesterday. We do know that the polycystins are part of a huge multi-protein complex, but the molecular components are not fully understood. And when we started this project many years back, we wanted to identify proteins in the polycystin-1 complex. And this all started with overexpressed protein, but Tilman then really took this to the next level by trying to investigate the native endogenously expressed polycystin-1 by endogenous tagging of PKD1 in IMCD cells with both N-terminal FLAG and C-terminal V5 tags. And with that, he was able to very efficiently immunoprecipitate different parts of polycystin-1 in these cells. Here you see the native PC1 interactome, and which was reassuring for us that the most abundantly co-purified protein with polycystin-1 was polycystin-2, so that was good. And then a whole range of additional proteins that were significantly co-purified. So we started doing pairwise co-immunoprecipitations to validate some of these, and here's just an incomplete list of some of these members. And the basic question was, can we learn something through these novel interactors about polycystin-1 processing and function? But to be honest, when we looked at this list, we weren't particularly inspired because as we all with these omics approaches, you get long lists and then it's difficult to make sense out of it. And in an ideal world, you have a secondary screen to prioritize your candidates. But since there are no good cell-based models for PC1 function, this should be done in vivo, which of course is very cumbersome. But luckily, nature did this screen for us and pointed to a role for DNAJB11 in polycystin-1 function. And this was done by the very nice paper by Emily Cornec-Legal at that time in Peter Harris' lab, who showed that monoallelic mutations of DNAJB11 cause autosomal dominant polycystic kidney disease. So that sparked our interest to look more into this PC1 interactor specifically. In this work and then some follow-up work in Emily's lab, it was shown that the prevalence of this disease was rather low, with a median age of end-stage kidney disease around 75 years. the cysts are much smaller and there's prominent interstitial fibrosis. And in fact, it was shown that the clinical features are somewhat of a mix of AD-PKD and AD-TKD. It was then later on shown in this follow-up study that there's mutations along the whole protein, and they were found in 27 pedigrees, so there's no doubt about the genetic association of this causing AD-PKD. At the molecular level, this was mostly studies in cell-based systems. DNAJB11 was shown to be a co-chaperone in the HSP70 cycle. It's a major cofactor of the ER chaperone BIP. And it has been proposed to have a central role in protein homeostasis, but there was very little known about its function in vivo. One question really that struck us and probably many others, why are there only cysts in the kidney if that protein has such a central role in protein homeostasis? And we set out to study this in more detail and try to address some key questions. First, what are the genetic mechanisms for the dominant inheritance of the disease? Which cell types drive cyst formation? And what are the molecular mechanisms of cyst formation? And we generated a mouse model, a cell model, and used some unbiased approaches. I'm talking a bit about our proteomics experiments today to understand better what's going on here. So we used a publicly available UCOM allele for DNAJB11, which through the use of flipase can be turned into a conditional allele, and then through Cre recombinase can be turned into a knockout. And the work on the mouse stuff was done mostly by Björn Neubauer, who's here to answer all your difficult questions on the mouse stuff. Björn first looked on the expression pattern of DNAJB11 using the LAC-C cassette and found that in early embryogenesis, DNAJB11 is already expressed pretty ubiquitously, including early kidney development. At later stages at P14, you find it in most cells of the kidneys, quite prominently expressed in kidney tubules. And Björn then went on to use this so-called TM1A allele, the so-called knockout first, to see whether he can get a phenotype if he generates a constitutive knockout. And what he found first, looking at the offspring, is that homozygous animals were way below Mendelian ratios. In fact, only one homozygous animal survived, so there must be embryonic or perinatal lethality. And when he looked at the kidneys of this one animal that survived, this was nice to see. It looked like the kidneys were indeed altered and they had a polycystic phenotype. So this motivated us to look further, and it's important to note that very similar data to a lot of the things that I'm going to report today were recently reported by Ghosh Roy in Whitney Bess's lab in a very nice JSON paper. But there's also some notable differences between our studies and additional data that I'm happy to discuss if you're interested. So Björn then went on to generate two alleles, one clean constitutive knockout by removing the neomycin cassette and the third exon, and then distal tubule specific knockout using KSP-Cre. And I start with the latter because we wanted to circumvent the embryonic lethality by using this kidney-specific knockout first. And for those who are not doing a lot of mouse work, just to remember that the KSP-Cre with a PKD1 floxed allele gives rise to a very striking cystic phenotype. So therefore, we were really surprised when we knocked out DNAJB11 in the distal tubule that the kidneys looked pretty much normal at P90. And that was true in the histology as well. You can zoom in and there's really no change in the tubular structure. And this was also true when we looked at 1 year of age. The kidneys really pretty much looked the same. And not surprisingly, there was no change in the kidney to body weight ratio, dilation or cystic index, and BUN was completely normal. So therefore then Björn decided to look at the constitutive knockout using a general deleter Cre, and again first looked at the Mendelian ratios. at E17.5, and already at that stage, there's a slightly decreased number of animals, as you expect from Mendelian ratios. And when he looked at P90, this was further decreased. So it's a combination of embryonic and perinatal lethality that gives rise to the demise of these animals. But once they survive the embryonic and perinatal period, animals can survive for a pretty long time. So Björn first looked at embryonic kidneys and found that wild type and heterozygous kidneys look the same, but cyst formation already starts during embryonic development. When he looked at adult kidneys at P90, he found that the kidneys are enlarged. In fact, he looked also whether this is truly a null allele, and the protein is completely absent in these kidneys, suggesting that we have a null allele at hand. The histology showed that there is massive cystic disease, although much less pronounced than in PKD1 deficient animals, and also interesting to note that the disease appears somewhat focal despite the constitutive knockout of DNA JB11. These animals, kidney to body weight ratio was increased in the homozygous animals, cystic index as well. Kidney function was still normal, which is always remarkable to me, how messed up kidneys can look and still have a completely normal BUN. So the next question was, which cells drive cyst formation, because of the surprising finding that when you knock out the gene in the distal tubule that you don't get any cysts, which was really unexpected. So we looked at nephron segment markers in wild type and knockout kidneys. LRP1 as a proximal tubule marker showed that the vast majority of cysts, almost all of them in embryonic kidneys, were derived from proximal tubules. When we looked at calbindin, a distal tubular marker, none of the cysts were stained with calbindin. And here you can see that in two colors, calbindin magenta, LRP2 in white. So there's really no cysts that are calbindin positive, and the majority of cysts are LRP2 positive. We also looked at the TAL by using UMOD. Again, no cysts are derived from the thick ascending limb or from the collecting duct, as shown by the aquaporin stainings. It's important to note here that there's no alterations in the thick ascending limb, and uromodulin levels appear to be normal. So this is not really in line with a connection to ADTKD, what the clinical phenotype suggests, at least at the molecular level. So in the adult kidneys, again, here you see a proximal tubule marker at P90. It showed that the majority of cysts were derived from the proximal tubule. There were few dilations of distal tubules that we observed in the adult animals, but this was really the minority. So Björn then went on to see whether doxycycline inducible knockout in all nephron segments gives rise to cyst formation, and used the doxycycline inducible PAX8 CRE to answer two questions. Whether loss of DNA JB11 in proximal tubules only triggers cyst formation, and whether postnatal inactivation affects the phenotype as it does for other cystic diseases. Here you can see the tubular knockout at P90, and there is no massive cystic disease, but the overall structure looks already a bit different in the tubule-specific knockouts. And if you zoom in, you can see that there's a very clear and widespread tubular dilation in the PAX8 animals. And this was highly significant. Again, as expected, no changes in kidney function yet. Björn then went on to look where these dilations are derived from. And again, like in the embryonic kidneys of the constitutive knockout, all these dilations were derived from proximal tubules. So going back to the initial questions that we asked, is the genetic mechanism for the dominant inheritance is recessive at the cellular level again, like in classical ADPKD. Heterozygous animals, at least to the time points we observe, do not develop any phenotypes. Which cell types drive cyst formation? It's proximal tubule cells, which was a real surprise to us. And does postnatal activation affect the phenotype? The answer to that is yes. It's a much, much milder phenotype, similar to other cystic diseases. So we also looked at fibrosis in these animals because that's a prominent feature in the human disease, and found that there's quite prominent fibrosis around cysts, shown here by acid fuchsin staining. And Björn also looked at smooth muscle actin, which stains blood vessels, but also fibrotic areas. And in the knockout animals, you see prominent fibrosis around the cysts, but no fibrosis in the non-cystic areas. And this was also kind of confirmed in the KSP-3 knockout animals, where there was no increased fibrosis compared to the wild-type animals. So next, we wanted to address the molecular mechanism of cyst formation and whether there's any functional interaction with polycystin-1, which we showed to physically interact with DNA JB11. One thing we're particularly interested in was GPS cleavage, which is known to be critical for PC1 function. And for those who don't remember this, one will briefly remind you of the beautiful work in Fang Xian's lab, who showed by knocking in a cleavage deficient mutant that the cleavage is really essential for polycystin-1 function, and cleavage deficient polycystin-1 gives rise to a cystic phenotype. And already in Emily's initial paper on the human paper, there was evidence that cleavage might be impaired in DNA JB11 deficient cells. So we wanted to look into that in genome-edited IMCD3 cells and generated DNA JB11 knockouts in these cells. Very importantly, we did this with isogenic controls. We wanted to generate null alleles, deleting the whole gene, and always looked at multiple knockout clones to be sure we had no clonal artifacts. And this is because we've learned our lessons with these clonal cells, and everyone working with these cells who has problems with clonal artifacts. I point to Luca's recent paper, where we found that the isogenic controls are really absolutely essential for these studies. To now study the cleavage of polycystin-1, this can be done with antibodies, and it's very easy to detect the N-terminal fragment with PC1 antibodies, with the 7E12 antibodies. It's more difficult to detect the C-terminal fragment. And we took advantage again of the knock-in cells that Tillman had generated with a C-terminal V5 tag, and generated DNA JB11 knockout clones in these cells as well. Here you can see a plot where we immunoprecipitated endogenous PKD1 with a V5, and you can see that there's two fragments, the full length, very little of it, and the C-terminal fragment in the wild-type situation. Now in DNA JB11 knockout cells, there's strongly impaired cleavage with a much more full-length product and very little CTF, and that's true in all of the independent clones. You can see the quantification of the C-terminal fragment over full length, which shows a highly significant impairment of polycystin-1 cleavage. So we then wanted to check whether DNA JB11 re-expression can rescue that phenotype and whether patient mutations have an effect on this. And here you see that re-expression of wild-type DNA JB11 gives rise to very similar levels to the endogenous level. This is a retroviral re-expression vector. We then looked at one mutant, a prolinto-arginine mutant at position 54, which gives rise to almost equal expression levels than wild type. And then we had a second patient mutation, which gives rise to very strongly decreased protein expression, which likely is a null allele. But we were particularly interested in using this mutant with equal expression to the wild type to see whether you can use PC1 cleavage as a functional readout for DNA JB11 function in variants of unknown significance. And here you can see the experiment we did to do that. Again, you can see the full length and the CTF impaired cleavage in the knockout. This can be rescued by re-expression of wild-type DNA JB11. when we expressed the mutant, which is expressed at normal levels, there's no rescue. And in fact, this looks exactly like the knockout cells. And here's the quantification of these data showing that you can indeed use PC1 cleavage as a readout for DNA JB11 function if you want to address functionality of the protein. Is this also relevant in vivo? Yes, we looked at PC1 cleavage defect in the DNA JB11 deficient mouse embryos and found impaired cleavage there as well. Here we looked at the N-terminal fragment and full length using the PC1 antibody, and there was also significant impairment of cleavage in vivo. So this shows that DNA JB11 is required for PC1 cleavage, but we wondered whether this is the only pathogenic mechanism, because the phenotypes and the cyst origin are quite different. And to this end, we used unbiased approaches. In this case, I'm showing you the proteomics data, and we compared the proteomics data from DNA JB11 deficient cells compared to wild type cells and the PKD1 deficient cells. We first looked at all these hits to see whether we believe that these models are good, and found indeed that there are quite a few variants that we could confirm
5 minutes
data. And one of them I'm just highlighting here, GLIS2, which you've heard a lot about yesterday. And indeed, our cell line, we believe, is a good model to mimic at least some aspects of the in vivo situation, because this recapitulates the strong upregulation of GLIS2. When we now looked at pathway enrichment using these differentially regulated genes, we found that the DNA JB11 deficient and PKD1 deficient cells showed quite different pathways that were most significantly enriched. And I don't want to go into the details, but just mentioning that these cells, while they may have some similarities, and PC1 may be a combining pathogenic factor, there's also quite a few differences. This is also highlighted here when you look at the overlap of the differential regulated genes, where you see that the more than 100 differential regulated genes, only 12 are commonly regulated in these cell lines. Some of them, like Col4A1, may be interesting candidates to follow up on when studying the impact of common pathways in these diseases. We also looked at other PKD genes, whether the proteins are differentially regulated, and found no other detected known PKD genes to be significantly misregulated in this data set. So for those of you who are interested to dive more into these data, you can look into the extensive data in the paper that is published in the meantime. And with this, I'm summarizing what I just told you. So the DNA deficient mouse model recapitulates major aspects of the human disease. DNA JB11 dependent cyst formation is recessive at the cellular level. Surprisingly, cysts develop from proximal tubules and not from distal tubule segments. DNA JB11 is required for GPS cleavage of PC1, and this was already shown in Whitney Bess's paper and is confirmed by us. We do think that the differences in phenotype, cyst origin, and proteomics data suggest that there may be additional PC1 independent disease mechanisms that are worth following up on. And if you think about future therapeutic implications, it's maybe worth considering that drugs targeting distal tubule-derived cysts like tolvaptan may not work if it turns out to be true that in the human disease, the major site of cyst formation is the proximal tubule. And for those interested in therapeutic approaches correcting, misfolded PC1, DNA JB11 is certainly an important player in PC1 maturation and processing. With this, I would like to acknowledge the people who did the work. Many people were involved in that, a whole range of MD students who did excellent work. But again, the two people who really spearheaded this project were Tilman Busch and Björn Naubauer. And with this, I thank you for your attention, and I'd be happy to take questions.
Thank you so much. I am going to dive in right away for the questioning part of it. We had lots of them coming in throughout you talking. So there is a lot of hype around why is there a proximal tubule phenotype and not the predicted or expected collecting duct phenotype. So I'm trying to summarize the questions here. One of the questions was, do you think that PC1 uses prominently a different protein for cleavage than DNA JB11 in the collecting duct versus proximal tubule, and that's why you see that? The other question was whether you think that potentially the proximal tubule is more sensitive in certain stages of development to the loss of polycystin than the collecting duct, and that's why you see that phenotype.
Yeah, so the short answer to these questions is I don't know. I'm not sure the proximal tubule uses other proteins for the cleavage, because the cleavage, from all we know from Fang's work, is an autoproteolytic cleavage. And our interpretation is that it's the correct folding and maturation of the protein that is required for the autoproteolytic cleavage. Whether you need additional proteins for this process that are expressed in the proximal tubule only, but not in distal tubules, or whether the requirement for GPS cleavage in distal tubule segments and the sensitivity is a different one, we don't know. But yeah, the combination of the segment markers with the complete absence of the KSP3 driven knockouts really suggests that, at least in mice, the proximal tubule is the prominent site of this formation. And yeah, it would be interesting to get a hand at, we don't have that at patient material at some point, to see whether that looks similar in the human disease.
Yeah, I would agree. The other question that came up is whether you looked at if DNA JB11 knockout affects polycystin 2 expression.
Yeah, the expression is not very strongly affected, but what is interesting is, and I didn't show you the data, and we've actually omitted it from the paper to make it not too complicated, the interaction between polycystin 1 and polycystin 2 is impaired in the DNA JB11 knockout. And that's probably because that is thought to be, the cleaved product is believed to be more critical. So the absolute level is not strongly impaired in our hands, but the interaction between PC1 and PC2 is strongly reduced in the DNA JB11 deficient cells.
Have you looked at the protein or the RNA level, which these days is somewhat easier to do with the various single cell data sets, whether DNA JB11 differs significantly among the various tubular compartments?
We've tried to look at it there, and it's not very clear-cut data, and also with the LAC-C. So there is one anatomy expert that claimed, looking at our LAC-C stainings, that it looks like it's more strongly expressed in proximal tubules. It's not so clear, I think. It's expressed really in all tubule segments. Whether there are slight differences in the expression level is hard to say. We've tried a whole range of antibodies, and we have a few that work really well on Western blots. Unfortunately, they all don't work for immunofluorescence, because in the knockout animals we get the exact same staining. So yeah, I don't think it's a strong difference for different tubule segments.
Two sort of ideas that go hand in hand coming from Dr. Parnell and Dr. Giamino. So the first question was, have you considered crossing it against the VV mouse? And the second part of that is from Dr. Giamino, stating that it is somewhat paradoxical of what you see, that DNA JB11 is responsible for PC1 cleavage, yet the VV mouse phenotype is predominantly restricted to the collecting duct. Whether you have anything there to say.
Yes, no, that was something that puzzled us as well, and we don't have a good answer for that. We would be great to cross it to the VV mouse. The problem here in Germany is that every modification of an animal protocol takes 6 to 10 months. We were already delayed after the KSP3 didn't work, and that's why we were so slow in publishing these data, because yeah, it takes ages to have any only slight modification for animal protocols to get them through. So yeah, it's nothing we're planning to do, but it is difficult to explain. And I think the impaired cleavage is a plausible mechanism for the cyst formation. I don't think that we have proof that it's the real cause. And I agree that it's paradoxical with the data from the VV mouse.
I think we're out of time. Thanks for the great questions that came in and for your answers. And we will move on to the next speaker.
Thank you.
So good morning, everybody. So it's a real pleasure to introduce Dr. Peter Harris. He is the Gordon H. and Violet Bartels Professor of Cell Biology, and he is the Zell Endowed Director of the Mayo Clinic Robert M. and Billy Kelly Perney Translational Polycystic Kidney Disease Center. He is a recipient of both the Kaplan International Prize as well as a Homer Smith Award. And among many things, his laboratory studies the genetic etiology of various forms of PKD. So today he'll be talking about the role of IFD proteins in monoallelic polycystic kidney disease. Thank you, Dr. Harris.
I believe you're muted.
Muted, yeah. Peter, are you muted?
Okay, can you hear now?
Yes, we can.
Okay.
Thank you.
How do the images look? Does it look okay?
Yes.
Okay, so I want to thank the organizing committee for inviting me to present work today. Some of it may be a little bit preliminary, but we'll see how it goes. So we're just to say I'm going to talk a little bit about the minor ADPKD genes and some of the mechanisms of action. Going to focus on IFT 140. And the disease associated with that particular gene. Look at some conditional knockouts we've done with the IFT 140, and then come back to think a little bit more about the penetrance of ADPKD genes and how population data can help us with those studies. So as you know, PKD1 and PKD2 are their major genes, accounting for about 78 and 15% of disease in a renal clinic population. There's some that are still genetically unresolved, some other genetic complexity, but there's a number of other loci that account for at least a proportion of these other cases, and I'm going to focus on those cases. So here are some of the genes that have been identified in the last few years. They tend to have a rather specific phenotypes, GANAB, a few cysts in the kidney, but sometimes severe polycystic liver disease. You've heard a lot about DNA JB11, so I won't talk more about that. ALG8 is mild kidney cyst. Typically, this is a rather atypical case. ALG8, we can see here cysts within the kidney, but severe polycystic liver disease is also possible. ALG5 probably has a phenotype somewhat like DNA JB11, and ALG6 I think is still rather speculative, but may also be more of a PLD gene. I took this kind of rather complicated figure from Emily Cornac-de-Gal's description of ALG5, just to show a central mechanism that's important in these proteins that I just highlighted, in that they're all in the ER here, and they're all involved in the glycosylation, folding, quality control, and trafficking of membrane and secreted proteins. And in red, you can see these different proteins either involved in glycan modification here, glycosidases here involved in the folding and quality control of proteins. DNA JB11 you heard about, the chaperone protein. And there's PLD proteins that are also involved in this process, involved in the transport of proteins into the ER. And the whole point of this process is to determine which proteins to send on through the Golgi and which ones to send for degradation because they're not properly folded. We showed with the description of GANAB that the generation of this mature form of polycystin 1 is greatly inhibited in GANAB minus cells. And this is more so than we see for other cells like E-cadherin or EGFR receptor. We can see that we see this reduction even in dosage reduction here, so heterozygous cells, as we see in patients. And this results in polycystin 2 not properly transporting to the cilium in GANAB cells. So we think that this whole quality control and folding and trafficking of proteins is how many of these proteins may be involved with PKD, although as you've heard for DNA JB11, it may be a little bit more complicated than that. So it's a bit of a surprise to us when we were doing genetic screening a few years ago on patients that were unresolved for PKD1 or PKD2 changes and found a number of in patients with pathogenic changes in the intraflagellar transport protein 140, IFT 140. You can see here there's a rather distinctive phenotype with a rather small number of large cysts seen biallelic. We showed segregation in families, indicating that it's really a monogenic disease. We had a fairly large cohort of 38 families with 59 individuals, and this gene accounted for about 2% of ADPKD patients, and these were ones that were newly screened, so maybe due to PKD1 or PKD2. If we look at the phenotype, that we can see that the kidney function is mainly conserved in these patients in red here, although there's a couple of cases with a decline in kidney function. The size of the kidney is quite variable, but it can be greatly enlarged here. And quite often it's indicated as a Mayo imaging class of atypical class A2A due to just because the enlargement's due to a very small number of cysts. We worked with John Sayer on the UK Biobank looking at this gene. And they were able to show that, in the PKD cohort within Mayo Clinic Biobank, truncating mutations to IFT 140 were the third most commonly found, and at that stage, the only other significant gene apart from PKD1 and PKD2. And consistent with that, loss of function variants compared to synonymous changes were found more often in patients identified with codes for cystic kidney disease, but not other kidney disease. This assertion that IFT 114 is a relatively important PKD, I think has been borne out by subsequent studies in the last 3 years. You can see this study from France here, identifying IFT 140, in this case, a rather small part of the population. This Japanese study showing 4.5% of the population. This Italian study here showing 2.3% of their ADPKD population. And then a recent large study by Emily Cadal indicating 2.7% of patients with ADPKD having changes in IFT 140. One point of note in that paper is that they're suggesting that there may be more decline in male individuals, and we'll need to keep an eye on that. One other study that's indicated the importance of IFT-140 for cystic disease is one from Natera, a genetic testing company. They found 368 individuals with pathogenic changes in IFT-140, and 60% of those were found in the group that were sent for testing because of a cystic phenotype. So suggesting a link with the cystic phenotype, although indicating that it may not be a fully penetrant disease. So it's not a surprise. I think that we all know that cilia is meant to be important in PKD. This just comes from polycystins and fibrocystin being present on cilia, but also syndromic forms of PKD, of ciliopathies often having PKD as part of their phenotype. So IFT-140 is the largest of the IFTA complex here that you can see. Is part of the core complex of IFTA, and it also interacts here with TOP3. This IFTA is involved with the retrograde transport of proteins in the cilium, although it's probably also associated with the regulation of import of proteins into the cilium. And there's a syndromic disease associated with this biallelically, which has a liver phenotype, a skeletal phenotype here. But in the original publication, kidney cysts and fibrosis were also marked as a part of the phenotype. If we knock out IFT-140 in completely in mice, this is a lethal ciliopathy phenotype, somewhat matching what we see in the human disease. In work that Greg Bazur's group did a number of years ago now, showing conditional knockout of IFT-140 in the kidney, we can see that it leads to a pretty rapidly progressive cystic disease here with many cysts by 20 days of age. So we've tried to do some inducible conditional knockout of IFT-140. This work done by Hannah Yang and Zoe Mann in my lab. And so using the PAX8 here, Cree, and knocking it out at different times, starting from E14.5 and P7, and then looking at the severity of disease at a later time. Of possible significance here, we used Doc's treated food and for 14 days to knock out the gene. So we can see here that if we knock it out at an early stage, 14.5, 15.5, 16.5, we get a pretty rapidly progressive cystic disease here. And we can see the total kidney volume here adjusted for length to look at the severity of disease here. And already by 16.5, it seems to be decreasing. Interestingly, in these studies, we didn't see much studies, and this is looking at 1 month in these images. We didn't see many cysts within the kidney. And this persisted really, this pattern. These animals are not viable till 3 months, but for the other animals up to 6 months of age, we saw very few cysts with the later knockout. The disease is progressive, though. If we look at the E16.5, or the ones with milder disease that survive to 2 months, we can see that there's a very rapidly progressive disease within the kidneys during that 1 month, and a doubling of the kidney volume. We can see also a similar pattern here if we look at the histology here for the E14.5 and 16.5, taken at slightly different times because we were aging these animals and sacrificing them before they died, but not many cysts within the P1 or P7. And you can see here the cystic index is more than 50% in these animals. If we do look now more carefully at the P7 at 1 year of age and at 9 months of age, you can see that there are a few cysts developing here within the kidney, but obviously not severe cystic disease. We've looked at the level of knockout here. This is the knockout product here, and you can see that this is the level E14.5 and the P7 knockout, which appear somewhat pretty similar to each other. It is a little lower at P1, and it may be that the DOX is not being transmitted well through the mother's milk, which may be accounting for a lower level of knockout. And so we're continuing these studies now, doing injections into pups to see if we see more aggressive disease in these lower ages, given the difference in data that we see here compared to what's been described for PKD1, PKD2, and for IFT 140. We're also want to develop a hypomorphic allele of IFT 140 and look at the cilia within that model. I want to transition now a little bit to talk about the frequency or penetrance of mutations in ADPKD genes. And we've taken advantage of the Mayo Clinic Biobank here. This is a large biobank, or about 54,000 individuals, which have whole exome sequencing data, but importantly also access to clinical records and imaging records, and about 50 to 70% of the individuals have imaging data. This is work but done here by DOA in the lab, and we started first looked at the penetrance of PKD1 mutations. There's been a lot of interest whether PKD1 can sometimes be silent. So in this case, we looked for tier 1, so truncating variants, or pathogenic or lightly pathogenic variants as defined in ClinVar. We found 77 individuals. 51 of these were in our ADPKD defined cohort. Of the other 26, 16 we felt were likely miscalls from the ClinVar. 9 were likely false positive truncating changes, and I'll show a couple of these. And there was one patient with a apparent canonical splice site that didn't have an ADPKD phenotype. But we can see here a high penetrance level. And we saw similar results for PKD2 here with 23 out of 24 with PKD2 changes in the ADPKD phenotype, and only one with a nonsense changes where we didn't have imaging data, apparently not having an ADPKD phenotype. So again, apparent high penetrance. These are a couple of the examples where one apparent nonsense change was associated with another missense change next to it, and so only results in a missense change, although the original call is for a truncating change. Likewise for this change here. And in another case, there's 2 apparent frame shifting mutations, but there you can see here that they're found in cyst with each other. So the result is a deletion mutation, which apparently seems to be neutral. So we went on to look at some of the other ALG genes or the other genes associated with ADPKD to see if they also what the penetrance of those are. So in a paper we've recently published in JASN, looking at follow-up of ALG8 and ALG9 in larger populations. we've been able to show here for ALG8 initially that we see kidney cysts in many of these patients and liver cysts, although you can see that some patients without kidney cysts or liver cysts with these variants. If we take a control population controlled for age and sex in the Mayo Clinic Biobank, we can see that the cyst number is higher than in the control population. We do the same studies in ALG9. There's less patients with ALG9. This is less frequent for these loss of function changes. We did see a trend in the right direction, but in this small population, it wasn't significantly higher for renal cysts in this group. But it's showing, I think, that there is not a complete penetrance here. If we look at the number of ALG8 variants in our ADPKD cohort, we can see only 2 out of 37%, so 5% would be diagnosed with ADPKD. And if we take a sort of minimal definition of ADPKD with more than 10 cysts, we can see that 50% would fit into that group. For ALG9, we can see that none actually fell into the ADPKD group, and 30% had more than 10 cysts in the kidney. So you can see for this gene, or these two genes, that the cystic disease is much less penetrant than we see for PKD1 or PKD2. We've also gone to look at IFT140 in the Mayo Clinic Biobank population here. Here there was 102 individuals with tier 1 variants, 43 of which had suitable imaging. We can see that most of them have cysts in the kidney. You can note that this is a somewhat older cohort, although there's some that didn't have cysts at all. But this was very significant compared to IFT, compared to the control population. Again, we see only a small minority of these being coded for ADPKD, but we see even less than 50% here having more than 10 cysts in the kidney. So despite the multiple studies indicating IFT140 is associated with a cystic phenotype, ADPKD phenotype, and the family studies, it's clear that it's not a completely penetrant disease where everybody with a variance in this gene has polycystic kidney disease. 5 minutes remaining. So I just want to highlight now on a slight diversion here, the monoallelic NEC8 disease that was described in 2023. This is a very interesting disease where just single changes give rise to this very rapidly progressive disease in the infancy, and often kidney failure within childhood. The mother here is a mosaic, so had milder disease. In work that Kun Ling here at Mayo has done, comparing cells re-expressing the wild type or the different variants that have been associated with the monoallelic phenotype. And I'll focus on this R45W because that's the most common. You can see that cilia were formed in these cells. The cilia length was not different from normal. The NEC8 was found on cilia in ANKS6, which interacts with NEC8, was also found in cilia here. But we can see that for polycystin 2, the level was reduced in cilia compared to re-expression of the wild type. And likewise in cells from patients, we saw lower polycystin 2 in cilia. So it's possible that the mechanism here is that the polycystin complex is not trafficking appropriately. Recently, we've turned our attention our attention was turned to another gene, IFT172, because we found patients with ADPKD phenotypes in our population screening. This is work we've done also with Emily Cornac-Legall. You can see here we found 12 individuals, unrelated individuals. We haven't really found families showing segregation at this time, but you can see that the cystic phenotype can be quite severe here, although it can also be quite mild, but suggesting a relationship with ADPKD. We looked in the, so IFT172 is an IFT-B complex protein here. The largest protein, which may be significant, we know large proteins like PKD1 and PKHD1 attract a lot of mutations. You can see the relative position of these two proteins in the IFT-A-B complex here. This is also associated recessively with a ciliopathy phenotype. If we look in Mayo Clinic Biobank, that we can see that these individuals often in green here develop cysts, although less so than for IFT140. Here's IFT140 for comparison, is IFT172, but nevertheless, it was significant compared to our control population. If we look at the ICD codes, we can see actually that 3 were in our ADPKD cohort for IFT172, but a rather relatively small level with more than 10 cysts. So I think a take-home message from these different other minor ADPKD genes is that they are associated with cysts in the kidney, but with a minor penetrance compared to PKD1 and PKD2. So just to summarize, if I have a few seconds, IFT140, it seems like the third most common ADPKD gene. We can see this distinctive phenotype associated with that. In higher preliminary loss of knockout studies, we're seeing only a phenotype with embryonic knockout. IFT172, another IFT gene, may be an ADPKD gene, although we need a little more data yet. And while PKD1 and PKD2 are fully penetrant, the IFT other minor genes are less penetrant. I've tried to thank people along the way, but Doa here, Hannah and Zoe in particular played a large role in the studies I described. And then I want to thank the other collaborators we have, both at Mayo and worldwide, that have played a role in this study. I want to thank Emily in particular that we've worked a lot with, and Cynthia has also played a big role in these studies. So thank you very much. Happy to take questions.
Thank you very much, Dr. Harris. That was great. So you have several questions already in the chat. So this one is from Feng Chen. Do you see the loss of cilia and IFT140 cystic kidneys? I'm assuming he means in the patients. And do IFT140 patients show cysts in the liver?
Yeah, unfortunately, because they very rarely reach renal failure, we've not got any data on, or we don't have any tissue from IFT140 patients, so we're not able to say what's going on. I mean, we're aware in the mouse models where we seem to see a kind of truncated cilia within IFT140, but we haven't had the material to be able to replicate that or see what's there in humans.
Okay, thank you. So from Chris Pedigo, have you studied PKD1, IFT140 double knockout animals? And do you see phenotype during adult inactivation?
Yeah, we would like to do that. One thing with IFT140 is that it's very close to PKD1. It's about half a megabase away. So we would have to make the mutation on, for instance, the RC background and then look at the disease phenotype, which is something that we would like to do, but we have not done that yet. We did breed together heterozygotes with IFT140 and PKD1 and PKD2, but we didn't really see an enhanced phenotype, even with the triple mutations there, or not very consistently anyway.
Okay, and then from Peter Charnecki, are the PKD-associated mutations in IFT 172 all C-terminal truncations, or do you see missense variants as well? The model would suggest a weakening of the IFT-A, IFT-B interaction upon truncation of IFT 172.
Yeah, we pretty much focused on loss of function variants in our analysis, so that there wasn't questions about whether missense changes were really pathogenic or not. So pretty much all of the ones that we've looked at would be ones that would truncate the end of the protein, and I mean, either the protein would get degraded away, or we would have a stable truncated protein.
Okay.
So that's a possibility for sure.
Okay. And I guess I have a question as well. Some of these IFTs, they can cause obesity, I guess, when they are homozygosed. So I was curious, in your patients, you see incomplete penetrance. So I was wondering if you see any obesity perhaps in some of your patients, and does that track with severity of cysts?
Yeah, we've looked for, with all of these genes, we've looked for other phenotypes that have been associated, with the ALG8 and ALG9. They're also associated with congenital disorders of glycosylation when biallelically. So we've looked for other phenotypes, and I have to say we've not really seen consistent phenotypes. Obesity is a tough one, I think, because there's quite a bit of it about. So I think it's difficult to tell whether it's significant. We haven't really noticed that, or other changes like skeletal changes. We haven't noticed that. So it doesn't. Those other phenotypes don't seem to be penetrant in a heterozygous way. And so why there's a different for the kidney phenotype than the others, I guess we're not really sure.
Okay. All right. I guess I'll just ask one more question. Do you see sex differences with your monoallelic IFT mutations?
Well, we hadn't noticed that actually, but in Emily's latest paper, she's suggesting that the males might have more severe disease than females. So it's probably something we should go back and look at, I think. I think in kidney size, we also not really noticed anything, but again, that's something we should probably review, I think.
Okay. Well, thank you so much, Dr. Harris.
No, thank you for allowing me to present. Thank you.
Okay. So we're going to move on to our next speaker, who is our selected abstract speaker. So this is Humaira Afrin from the Mayo Clinic. And is she here? Yes, she is. Okay, perfect. And so the title of her abstract is Kidney Organoids Demonstrate that the Stimulatory G protein coupled receptor parathyroid hormone type 1 receptor drives a cyclic AMP, PKA, PKREB axis in developmental polycystic kidney disease. Thank you.
Can you see my screen?
Yes, we can. It's just not in presenter mode.
Is it now?
No, not yet.
You need to swap your displays up at the top, middle. It says, yeah, it says swap displays. There you go.
Okay, thank you.
There you go.
Thank you for introducing me. I'm Humaira Afrin, working as a postdoctoral researcher in Dr. Gupta lab in the Division of Nephrology and Hypertension. I will be presenting our work entitled Kidney Organoids Demonstrate that the Stimulatory G protein coupled receptor PTH1R drives a cyclic AMP, phospho-PKA, and phospho-PKREB axis in developmental polycystic kidney disease. By developmental polycystic kidney disease, we mean ARPKD. So in the beginning, I would like to give a brief background of the kidney organoid. So here we are using Morrison protocol to develop the kidney organoid. And in Morrison protocol at day 8, we have the high efficiency of 6-2 induction, and then we make the organoid from it. Just to tell that the cells we are using is, we received it from the PKD RRC. It is PGP1 PGP1 and its CRISPR mutant PKHD1 double knockout. So at day 21, we do a complete QC to see the nephron structure of the organoids, which includes CDH1 distal tubule and LTL positive proximal tubule. So with the organoid that passed the QC, we do the experiment only with that batch of organoids. So after that, we know from the previously published study that suspension culture is a factor for the formation of the cyst. So just to confirm, we added forskolin in the adherent culture, both in wild type organoid and also in the PKHD1 double knockout organoid. But we did not see any cyst formation even after adding the forskolin in the adherent culture, nor in the control organoid. However, when we added the forskolin in suspension culture, we did see cyst formation. And to confirm the same organoid, we stained and we noticed that the cyst was formed in the LTL positive proximal tubule and in the distal tubules. So in the before, we added high dose forskolin, and then to minimize that, to see the dose adjustment, we found that forskolin produce a dose dependent cystogenesis, and with the lower doses for higher duration, it produced modest amount of cystogenesis. And in the bottom side, you see that we have stained those organoid and to see that where is those dilatation coming from. In the bright field microscopy, you can see this ovoid lucency that corresponds with the cystic dilatation. So as a confirmatory immunostaining, we see that the dilatation is coming from both the proximal tubule and the distal tubules. So by now we know that the forskolin induces the cyclic AMP, and that activates the downstream pathway of cystogenesis by phospho-PKA and phospho-CREB. So we wanted to know what are the other pathway that would stimulate the G protein couple receptor and would activate the downstream pathway. So we did an unbiased screening in the organoid to see to find out the other GPCR receptors, and we found that PTH1R and PTH2R both are present in the kidney organoid. However, these are not present including arginine vasopressin. So now we know that PTH1R and 2R are present in the kidney organoid in the mRNA level. Does it present in the protein level in the organoid? So to see that, we stained those organoid for PTH1R and 2R, and we found that PTH1R is abundantly present in both in the proximal and distal tubule, whereas PTH2R is not that abundantly present. It is very negligible amount present in the tubules in kidney organoid. So by now we know that the kidney organoid has PTH1 and also a negligible amount of PTH2, but does it present in the human? So we interrogated the data available at the KPMP website, and we found that both the PTH1R and 2R is present in the throughout the nephron. But this is for the healthy reference. Is it present in the cystic patient or the cystic kidney? Then we received the AR-PKD cystic kidney sample from the PKD RRC. Then we stained it for the PTH1R and PTH2R, and we found the presence of PTH1R and PTH2R both in the tubules. And so now that we know that PTH1R and PTH1R is present in the cystic kidney, we want to know, is it driving the cystogenesis? So it came from a simple thought that we know that from the clinical data, we know that PTH is inversely proportional to the kidney function, and the kidney volume is also inversely proportional to the kidney function. So if A is to B and B is to C, so perhaps it is true that A is to C. And also from the clinical data, we know that PTH is directly related to the total kidney volume, and in the ADPKD patient, irrespective of the kidney function. So to confirm our hypothesis, we treated the kidney organoid with different doses with PTH and PTH-RP. The PTH works in both the PTH1 and 2 receptor, and PTH-RP, it only works in the PTH1 receptor. So we found that both the PTH and PTH-RP, it induces cystogenesis. We can see in the bright field by the ovoid lucencies, you can see those cystic dilatation. And to confirm, we fixed and cut the section and then stained it, and we found that, and we found dilatation in both in the proximal and the distal tubules. So now we know that the PTH is inducing a cystogenic effect. So we want to know what are the downstream pathway. Is it inducing cystogenic effect through adenylyl cyclase, phospho-PKA, and phospho-CREB axis or not? So in the beginning, we stained the kidney organoid treated with PTH to see that if there is a phospho-CREB or phospho-PKA substrate. So after staining, we found that it does have the phospho-CREB and both phospho-PKA substrate. And then we think, okay, if we inhibit the adenylyl cyclase or PKA inhibitor, will it reverse the cystogenesis? So then we treated the organoid and induced the cystogenesis with PTH, and then treated them with adenylyl cyclase inhibitor and phospho-PKA inhibitor, and with different doses. And we found that at the higher dose, both reduced the formation of the cyst and reduced the size of the organoid. But again, to confirm, we stained and we found that in both of the treatment, and the LTL and CDA, the proximal and distal tubule size got reduced. So in summary, I would like to say that from our study, we found that the suspension culture and forskolin both act as a cystogenic stimuli for an AR-PKD organoid. The PTH receptors, PTH1R and PTH2R, are stimulatory G protein coupled receptors that are present in the kidney organoid, native kidney, and also AR-PKD kidney cyst. And PTH1R agonism provide a cystogenic stimuli to AR-PKD organoid, and PKD induced cystogenesis independent of an AC, phospho-PKA, and phospho-CREB axis. With that, I'd like to thank my lab, my whole lab, and Mayo PKD Center, particularly who does the basic science, Dr. Harris, Torres, Lee, and Hugh, and PKD RRC, not only to provide a couple of the resources of this study, but also to provide the funding of this study. Thank you.
Thank you very much for the excellent talk. Do you have any idea of how PTH1R signaling is upregulated in ARPKD?
The PTH, the PTH1R is how it is upregulated in ARPKD.
Yeah, that was the question.
The PTH1R is upregulated in ARPKD. We can see it. I don't know exactly how, but some of it comes from the secondary hyperparathyroidism. So in patient, when there is secondary hyperparathyroidism, there is the imbalance of the calcium, and to adjust that, there is the upregulation of the receptors.
Okay, I see. Given the known role in kidney physiology of PTH, Dr. Wallace asked whether it is all about just driving fluid secretion or whether there's actually a direct role for PTH to increase ERK activation and proliferative positivity in the cells or in the organoid.
That's an excellent question that we would like to explore more in this area. We still have not studied it yet. Thank you.
The next question, or the other question that was put, whether you have any explanation why the V2 receptor is not expressed in the collecting duct within your organoids?
Yeah. So the organoid we are using, it's the nephron organoid. It does not have the collecting duct structure. So, and the arginine vasopressin is present in the collecting duct. As we do not have that structure, so it does not have that receptor there.
Okay. Are there antagonists that you can use that are otherwise used in the field for PTH signaling to reduce the cystogenesis?
We only used adenylyl cyclase inhibitor and PKA inhibitor. That is the two inhibitor we used to reverse the cystogenesis.
Thank you. The other question that I have is, have you translated any of these findings or looked in the AD-PKD realm?
That is an excellent question. We do have the we do have the patient sample of AD-PKD, and we did the spatial transcriptomics on both the samples to find out the convergent and the divergent pathway of the cyst formation of those, and that is something definitely we're gonna work in next.
Yeah, I think it would be interesting to see whether the increased levels as either correlate, just like seen for ARPKD, or whether it's an avenue of treatment, or whether with PTH you can at least exacerbate the disease.
Yeah, we will definitely look at that.
Okay, thank you very much again. It
Thank
seems like we covered all the questions in the chat. I think people must be ready for lunch, but excellent talk. Thank you.
Thank you.
All right, I want to thank the speakers for this session. Outstanding talks. We will have a lunch break until 1:30. Terry, do you have anything that you wanted to add? Are you?
No, I just want to thank everybody. That was really great morning.
See everybody at 1:30.